Hello everyone and welcome to my presentation. My name is Francisco Cajas and today I'm thrilled to present my scientific poster on one of the most revolutionary advancements in modern genetic engineering, and that is recombinant DNA technology. So let's start by discussing its purpose. What problems does it solve and why is it so important? Well, before this technology was developed, obtaining therapeutic proteins like human insulin or growth hormones was incredibly difficult, expensive, and often unsafe. For instance, insulin had to be extracted from the pancreases of slaughtered pigs or cows, which frequently caused allergic reactions in human patients. That's when recombinant DNA technology entered and solved this by allowing us to take a specific gene from one organism and insert it into the DNA of another, usually a bacteria. This forces the host organism to act as a biological factory, producing valuable substances efficiently, safely, and in massive quantities. It is a cornerstone of modern medicine, agriculture, and academic research. So let's start with the materials and components. Before we look at the process, let's meet the essential tools and molecular machinery required to make this happen. If you look here we have several key components. First, the target gene, which is the specific sequence of DNA we want to copy or express, such as the human insulin gene. Next is the bacterial plasmid, which acts as our vector. Plasmids are small, circular, double-stranded DNA molecules, distinct from a bacterial chromosomal DNA. To manipulate this, we use two critical enzymes. Think of restriction enzymes as our molecular scissors. They recognize and cut specific DNA sequences. Then we have the DNA ligase, which acts as our molecular glue, sealing the DNA backbone together. We also need a bacterial cell like E. coli to act as our host to replicate the new DNA. Lastly, we use an antibiotic resistant gene as a selectable marker, and obviously lab equipment like agar plates to screen our results. So let's go through the step-by-step process. Now let's walk through the step-by-step procedure of how we actually construct and express recombinant DNA. There are six fundamental stages. The first one is isolation. Here we extract the total DNA from the donor human cell containing our target gene, and simultaneously we isolate the circular plasmid from the bacterial cells. The second stage is cleavage. This is where our molecular scissors come into play. We treat both the human DNA and the bacterial plasmid with the same restriction enzyme because they are cut by the same enzyme. They both develop complementary single-stranded overhanging ends known as sticky ends. As a fun fact, if we use different enzymes, the ends wouldn't match and the process would entirely fail. The step three is ligation. Here the cut we mix the cut plasmids and the target human genes together because of their complementary sticky ends. They naturally base pair with each other, and then we add DNA ligase. This enzyme restores the phosphodiester bonds in the DNA backbone, permanently gluing them into a single unified loop. We now have a brand new molecule called a recombinant plasmid, and there is where stage 4 enters transformation. Now we need to get this recombinant plasmid back into a living bacterial cell so it can replicate. Bacteria don't normally just soak up in foreign DNA, so we have to force them. We do this by treating the cells with calcium chloride and giving them a sudden heat shock. This temporarily creates pores in the bacterial cell membrane, allowing the plasmid to slip inside. And the step 5 is cloning and selection. This is crucial quality control step. Not every bacterium will successfully take up a plasmid during transformation. To find the ones that did, we grow the bacteria in a selective agar medium containing an antibiotic. Remember that antibiotic resistant genes we included in the plasmid? Only the bacteria that successfully absorb a recombinant plasmid will survive and grow into colonies. The unsuccessful ones will die. The final step is expression and harvesting. The surviving bacteria are transferred to large bioreactors where they multiply exponentially. Inside these cells, the cellular machinery automatically transcribes the recombinant DNA into mRNA. and translates it into the desired human protein. We then rupture the bacteria, purify the protein, and harvest it for clinical use. Now let's go for applications in the real world, in real world applications. Because this technology isn't just theoretical, it has completely transformed our daily lives. Let's look at two major real world applications. First, in medicine, it is used for producing human insulin. Millions of people worldwide suffering from type 1 diabetes rely on daily injections of humalin, which is human insulin manufactured entirely by recombinant bacteria. It is pure bioidentical to what our own bodies make and completely sustainable. The second example is in agriculture. It is used to create BT corn. Scientists took a gene from a natural soil bacterium called Bacillus thuringiensis, which proceeds as a protein toxic only to specific insect pests, and inserted directly into the corn genome. As a result, the corn protects itself from the pests, drastically reducing the need for chemical pesticides, which protects our environment. Now, the connections to genetics and the conclusion. To wrap up, how does this technology connect back to everything we have studied in our genetics unit? When it comes to DNA technology, is the ultimate proof of the universality of genetic code. The fact that a bacterium can read a human DNA sequence, transcribe it, and translate it into a perfectly functional human protein proves that all living organisms share the same fundamental genetic language. It directly applies the core principles of the central dogma of molecular biology. DNA makes RNA via transcription, and RNA makes protein via translation. Without understanding DNA replication and gene function, this technology could never exist. All the information presented today is backed up by five reliable academic sources listed here at the bottom of my poster, including molecular biology of the cell. Thank you so much for your time and attention. I hope this video gave you a clear and engaging insight into how we write the code of life to solve global problems. Uh, I can read you in the comments for any questions.